Hello, Namaste. I'm Dr. Amit. So today we'll continue our neuroanatomy lecture, and today we'll talk about different types of glial cells. Okay, so we know that we have ependymal cells, microglia, Schwann cells, oligodendrocytes, satellite cells, and astrocytes. Okay, so we'll talk about the special features of all these different types of glial cells. So let's start our lecture on different types of glial cells. So before starting the lecture, let's see this structure once. Okay, so this is a structure of neuron. Okay, so this is cell body, and you can see this is cell membrane, and this outward coming part is dendrites, and this is axon. Okay, and over axon we have this myelin sheath. Okay, this blue, and this yellow is axon. Okay, and in between you can see the gap. Okay, this gap is nodes of Ranvier. Okay, and this myelin sheath is secreted by oligodendrocytes in central nervous system and Schwann cells in peripheral nervous system. We'll talk about that. Okay, and this is axon terminal, or we can say that synaptic end bulbs. So first we'll talk about astrocytes. Okay, so this astrocyte is derived from neuroectoderm. Okay, remember this neuroectoderm. The astrocyte derived from neuroectoderm because they ask in this line in MCQs. Okay, and it forms a blood-brain barrier. We all know there is a barrier between brain and blood. Okay, so this astrocyte helps in formation of blood-brain barrier, and the shape of this astrocyte is star shaped. Okay, and this astrocyte is the largest and the most abundant glial cells in our central nervous system. Okay, and while talking about its functions, okay, it helps as a physical support. Okay, so it provides physical support, and it also helps in repair. Okay, if any damage in the CNS, and it helps in repair, and it also helps to remove excess neurotransmitter. Okay, whenever there will be excess neurotransmitter in our brain, then there will be overstimulation and it will cause damage. So the astrocyte help us to remove the excess neurotransmitter in our brain. Okay, and it also act as a glycogen fuel buffer. Okay, so whenever there will be low glucose in our body, or whenever there will be high demand of glucose in the body, then this astrocyte act as a reserve. Okay, it act as a reserve for fuel. Okay, now while talking about reactive gliosis in response to neural injury. Okay, so what this line means that whenever there is damage to the CNS, either by infection, stroke, or tumor, okay, so these astrocytes will proliferate. It means it will increase in number, and it will form a protective barrier or a scar, okay, to that neural injury, and it will cause a scar. And this is a reason while there is that in PNS the axonal regeneration is very good, but in CNS the axonal regeneration is not so good, because this astrocyte will form a scar, okay, neuronal scar after reactive gliosis. And while talking about its lab property, it's GFAP positive. It means that glial fibrillary acidic protein, okay, in immunohistochemistry it is positive for GFAP. Now we'll talk about microglial cells. So microglial cells are derived from mesoderm. Remember this line. And microglial cells are actually the macrophage of the CNS. Okay, so that's why it's a phagocytic scavenger cell of CNS. Okay, now whenever there will be tissue damage, then there will be activations of microglial cells. Okay, so to release the inflammatory mediators like interleukin-1, tumor necrotic factors. Okay, and then they release inflammatory mediators. Okay, and here is no glutamate, because glutamates are released by neurons, not by microglial cells. Okay, so they only release inflammatory mediators. So when there will be HIV infections, okay, in any person, then it will infect microglial cells also. So many infected microglial cells in HIV infected persons or patient, and will form a multinucleated giant cells in CNS. Okay, and that will lead to HIV associated dementia. Okay, so simply that microglial cells will show immune response, and it will release interleukin-1, tumor necrotic factor, and remember that it's derived from mesoderm. Now we'll talk about ependymal cells. Okay, so ependymal cells are derived from neuroectoderm. Remember that it is derived from neuroectoderm, and these ependymal cells are ciliated. Okay, ciliated simple columnar glial cells. Okay, and basically these ependymal cells lines the ventricles and central canal of spinal cord. Okay, so and over there it helps to in the production of cerebrospinal fluid. Okay, so and these ependymal cells apical surface are also covered by cilia, and that cilia will help to circulate the cerebrospinal fluid, and it will help in CSF absorption also. So while talking the important points about ependymal cells, just remember that it's derived from neuroectoderm, and it helps in productions of cerebrospinal fluid. Okay, it helps to produce CSF, and it lines the ventricles. Now we'll talk about myelin. Okay, so I've shown you in the picture also, okay, that this the myelin sheath or myelin is something that will increase the conduction velocity of signal transmitted down the axons. Okay, and at nodes of Ranvier there will be more sodium channel concentration, so there will be saltatory conduction. Saltatory conduction, simply just remember that that I'll show you the picture. So we all know, okay, these are the myelin sheath, this blue, and this is nodes of Ranvier, this gap. Okay, so when the signals will come, okay, through the axons, okay, so it will travel fast in this myelin sheath, okay, and its nodes of Ranvier there will be saltatory conduction. It means that the signal will jump from this myelin sheath to this myelin sheath at nodes of Ranvier, and this is known as saltatory conduction. So it's written here that saltatory conductions of action potential at the nodes of Ranvier. Okay, now remember this line, okay, that in central nervous system we know we have brain spinal cord, in peripheral nervous system we have that cranial knobs. Okay, so in central nervous system this myelin is synthesized by oligodendrocytes, and in peripheral nervous system it is synthesized by Schwann cells. Okay, now we have also have a mnemonics as COPS, that in central nervous system oligodendrocyte, and peripheral nervous system Schwann cells. Okay, now when the myelin wrap, okay, suppose this myelin wrap on axons, okay, then what will happen? Okay, now we'll talk about this. So first, it will decrease the membrane capacitance. Okay, so think that membrane capacitance is simply that myelin sheath will act as an insulator, so the membrane stores less charge. Okay, so when the membrane stores less charge, then it will decrease the membrane capacitance. Now while talking about membrane resistance, okay, so just remember that resistance means how much ion leakage is blocked. Okay, so more ion leakage will be blocked, that's why more it will increase the membrane resistance. Now while talking about a space constant, okay, so a space constant, just remember that how far electrical signal spreads before it decay. Okay, so it will increase because the signal will move more farther distance, okay, before it decay. Now while talking about the time constant, okay, so it simply mean that how fast membrane voltage changes. Okay, so it will change more fast, so that's why it decreases the time constant. So in MCQs they can ask this. Okay, so just understand these terms, and you can solve the MCQs. Now let's talk about Schwann cells. Okay, we all know that Schwann cells helps in the productions of myelin in peripheral nervous system. Okay, so it helps in promoting axonal degeneration. Okay, so axonal degeneration is very good in peripheral nervous system than CNS, because in CNS astrocyte will produce scar cells. Okay, that why the regeneration will be not so good, but the Schwann cells will regenerate axon more better in peripheral nervous system. Okay, and it is derived from neural crest cells, and these Schwann cells are actually injured in case of Guillain-Barré syndrome. And while talking about this cell marker, remember this S100 is a cell marker of Schwann cells. Okay, so while talking about Schwann cells, remember that in Guillain-Barré syndrome it injured, and its marker is S100, derived from neural crest cells, and it helps in promoting axonal regenerations. Now at last we'll talk about oligodendrocytes. Okay, so we all know that oligodendrocytes helps in productions of myelin synthesis in central nervous system. Okay, so it's written here that myelinates axon of neuron in CNS. Then each oligodendrocytes myelinates many neurons, many axons, not only one axons. Okay, then it's a predominant type of glial cells in white matter. So we'll talk about what is gray matter, what is white matter. Okay, so in white matter the oligodendrocytes will be the predominant type of cells. Okay, and it's derived from ectoderm, and histologically it appears as a fried egg. Okay, I'll show you the picture. Okay, and while talking like Schwann cells get injured in Guillain-Barré syndrome, oligodendrocytes get injured in multiple sclerosis, progressive multifocal leukoencephalopathy, and leukodystrophy. Okay, remember these three diseases. So histologically these oligodendrocytes look like a this fried egg appearance. Okay, so you can correlate this. Okay, So this is all about our different type of glial cells, that is astrocytes, microglial cells, ependymal cells, myelin, Schwann cells, and oligodendrocytes. So if you like this video, please share with your fellow medicos, and please subscribe my YouTube channel at the rate Dr. Amit Official, and we'll meet in next video of neuroanatomy. Thank you.